Hey everybody! We're now going to learn some more details about karyotyping, that process we learnt about earlier. So remember, karyotyping is it's the process of making a karyogram or a karyotype. And they show the same information. The karyogram is the visual diagram of the chromosomes, and the karyotype is just the written form of it, the description. So here is the process of how scientists actually make these karyograms. The first step is they take out a blood sample from the patient. This is the method for humans. It's different for plants and for other species like fungi, but this method, it's the same for humans and for other animals that have blood, which is most animal species. So we'll call it karyotyping animals. So you take a blood sample because blood contains cells, and some of those cells have a nucleus. Remember, red blood cells don't. They're just there to carry oxygen and they lose their nucleus. But white blood cells, they've got a nucleus, so we can get the chromosomes from those white blood cells. So they take the white blood cell, then they get a microscope slide, and they spread out a drop of blood onto that slide by dropping it from a bit of a height. The next stage is, you can see here, they digest it and then they stain it. So the digesting is there with trypsin, and that's a protein, and that protein breaks down most of the cell, but it doesn't break down the DNA. So it breaks down the cell and it lets the chromosomes spill out of the nucleus. And then they also stain it to make the chromosomes visible as well. So once they've done that, then they put that slide under a microscope and they zoom in on one particular cell and they look at all those chromosomes that had spilled out of the nucleus of that cell. So they find that one cell, they take a photo of it, and suddenly they've got a microscope image of all the chromosomes that you can find in that person. And from there, they just take the photo and use Photoshop or some other software to cut and paste each chromosome, put them in order, and arrange them neatly in a karyotype. So people actually have to do that because chromosomes don't just line up themselves in a neat karyotype. So the last step is to rearrange them. And from there, you can go on and diagnose if that person has a genetic disorder or not to do with their chromosomes. The common question that gets asked is, why do they stain the chromosomes? Well, I said earlier, it helps to make them visible. That's true, but it also helps scientists to figure out the chromosome pairs. So it makes the chromosome pairs visible. And that's because you can see here, each particular chromosome has a particular pattern of bands on it. You can see this chromosome here has dark, then a light, then a dark band and a light band, dark and light again. So each particular chromosome gets stained slightly differently, and that makes it really easy to tell which two chromosomes need to match up into their pairs. So each pair has a characteristic pattern of bands on them. So that is the basic process for how we do karyotyping.